Imagine your immune system is like a highly trained military.
But like an elite defense force.
Exactly. And most of the time, it operates with absolute precision. You know, you get a virus, the troops deploy, they neutralize the threat, and then they stand down.
They pack up and go home.
Right. The system works because the chain of command is totally intact. But what happens when the commanding officers, and specifically here we're talking about a highly specialized group of cells called T follicular helper or Tfh cells, what happens when they just go rogue and start ordering artillery strikes on your own healthy tissue?
I mean, it basically becomes a biological civil war.
Yeah.
That catastrophic breakdown in the chain of command, that is the defining feature of severe autoimmune diseases like systemic lupus erythematosus. The immune system's precision essentially just collapses, and the troops are actively directed to destroy the host.
Which brings us to the source material we are looking at today, because it offers a genuinely groundbreaking perspective on how to stop that civil war.
It really does.
We are jumping into a 2026 paper published in the journal Immunity by Wang and colleagues. And the mission of this deep dive is to explore how a common, everyday medication, like literally a statin you might already be taking for your cholesterol, might hold the secret to stopping these rogue commanding officers by cutting off their microscopic supply lines.
It's a fascinating mechanism.
Okay, let's unpack this, because before we get to the statins, we need to understand the commanders themselves. What exactly is a TFH cell doing on, a normal day?
Well, to understand the trouble they cause, you have to look at their normal day job inside your lymph nodes. So TFH cells orchestrate this microscopic environment called the germinal center.
Okay, the germinal center.
Right. Think of the germinal center as an elite boot camp. And the TF cells, they are the drill sergeants. They physically interact with B cells, which are the soldiers that actually manufacture our antibodies, and they help those B cells mutate, refine, and produce these highly targeted antibodies to fight off infections.
So they're basically handing out the target coordinates.
Exactly. Without TFA cells giving those orders, our B cells would just produce weak, completely ineffective antibodies. I mean, vaccines literally wouldn't work. We absolutely depend on them.
But in a disease like lupus, that boot camp becomes corrupted, right?
Yeah, the drill sergeants get completely confused. Instead of training the B cells to attack a virus, they start telling them to make autoantibodies.
So antibodies designed to attack the patient's own DNA, their own kidneys, their skin.
Precisely.
Uh-huh.
Which has left researchers just, banging their heads against a wall for years asking, how do we shut down these hyperactive, rogue TFA cells without carpet bombing the entire immune system?
Because if you wipe out all the T cells, the patient is left defenseless against just common infections.
Right. And the answer, according to this new research, lies in a cellular pathway called the mevalonate, or MVA pathway.
Yeah, the MVA pathway.
Yeah. Conventionally, the MVA pathway is famous in medicine for one major thing, manufacturing cholesterol. It is the exact pathway that statin drugs target to lower your risk of cardiovascular disease.
But the researchers found a massive plot twist here. The rogue TFH cells driving lupus, they aren't addicted to the cholesterol itself.
No, the cholesterol is basically irrelevant to their malfunction.
Right. Instead, these immune commanders are secretly addicted to a totally different, somewhat obscure byproduct of this MVA pathway. It's a molecule called geranyl diphosphate, or GGPP.
GGPP, yeah.
And when the researchers blocked the specific pathway that makes GGPP, the rogue TFH cells didn't just die. They essentially lost their identity, lost their ability to communicate, and just stopped functioning.
Yeah, to understand why blocking GGPP disables these rogue cells so completely, we have to look under the hood at the cellular machinery. We need to look at what this obscure GGPP molecule actually does inside the cell's factory.
So if the cell is a factory, the MVA pathway isn't just churning out cholesterol as its main product, it's also running the side assembly line manufacturing GGPP.
Exactly. And the paper breaks down the exact cast of characters here. There is an enzyme-like, a biological catalyst called GGPS1, and its entire job is to manufacture this GGPP.
Okay, GGPS1 makes GGPP.
Right. Then another enzyme steps in called GGTase II. You can think of GGTase II as a highly specialized factory mechanic.
A factory mechanic, I like that.
This mechanic takes the newly synthesized GGPP and physically attaches it to a specific group of proteins called RB proteins. And in this paper, the researchers narrowed the focus down to one RB protein in particular, which is RAB35.
RB35.
Yeah. And the process of the mechanic attaching the GGPP to the RAB35 protein is called granulation.
I mean, I love the biological jargon, but let's make it really visual for anyone trying to picture this.
Yeah.
The MVA pathway produces GGPP. Let's visualize GGPP as highly specialized microscopic screws.
Microscopic screws, perfect.
And the mechanic, GGTase II, uses those specific screws to assemble the RAB35 machinery. Without those screws, the RB35 machine just falls apart.
It can't function.
But wait, if RAB35 is just a piece of cellular machinery, why does a TFA cell care so much about it? I mean, can't the cell survive without one specific machine? What actually happens to the cell if it doesn't have those screws?
Well, answering that requires us to look at the outer surface of the TFA cell. TFA cells have surface proteins that act like sensory organs.
Okay.
The most critical one for their job is called CXCR5. Think of CXCR5 as a highly sensitive cellular GPS antenna.
A GPS antenna?
Right. It detects a specific chemical beacon called CXCL13, which is essentially a homing signal that guides the TFA cell through the crowded lymph node directly toward the B cells so they can interact.
So if you don't have that GPS antenna on their surface, they just can't navigate. They can't find the B cells to issue their destructive commands.
Exactly. And here's the critical mechanical mechanism the researchers uncovered. Those CXCR5 antennas aren't just glued to the outside of the cell permanently.
No, they're not.
No, the cell is constantly pulling them inside in a process called endocytosis, evaluating them, and then sending them back to the surface to keep the signal fresh. It's a continuous, rapid recycling loop.
Wow, okay.
And RAB35 is the engine of the transport vesicle, the recycling truck, that drives the CXCR5 antennas from inside the cell back up to the surface.
Okay, I want to push back on this concept for a second. If RAB35 is just a recycling truck, why does stopping it neutralize the entire cell? Couldn't the TFF cell just, I don't know, manufacture brand new CXCR5 antennas and stick them on the surface instead of relying on recycled ones?
It comes down to metabolic cost and speed. Manufacturing brand new complex surface receptors from scratch takes significant time and energy.
Right, that makes sense.
In the really fast-paced environment of an immune response, the cell has to dynamically adjust its sensitivity to chemical gradients second by second. Recycling is instantaneous.
Ah, so it's a speed issue.
Yeah. If you stall the recycling truck, the existing antennas get pulled inside the cell and trapped there. The surface is stripped bare almost immediately, long before the cell could ever synthesize replacements.
So let's do a quick recap. We're about 5 minutes in. Let's put the whole assembly line together. No GGPP means the mechanic doesn't have the screws.
Right.
Without the screws, the RAB35 recycling truck engine stalls.
Exactly. And the researchers proved this physically using immunoelectron microscopy.
Oh.
Yeah, they literally shot electrons at the tissues to visualize the nanoscale physical location of these proteins. They saw RAB35 and CXCR5 co-localizing, so physically stuck together in the same tiny transport vesicles near the cell membrane.
That's incredible. They can see that.
It really is. And to confirm it, they used genetic knockout lice, engineered so they could delete the gene for the GGTase II mechanic specifically in T cells.
So they just removed the mechanic entirely.
Right. And when they did, flow cytometry showed a massive, precipitous drop in CXCR5 on the surface of the TFA cells.
But the total amount of the antenna protein in the cell didn't drop, right?
Correct.
It just got trapped inside the cellular garage. The recycling truck stalled, the antennas were stuck, and the TFE cell essentially went blind. It couldn't navigate, and the autoimmune chain of command was broken.
That's exactly the mechanism.
Okay, so if I'm tracking this right, the cell manufactures these GGPP screws, which builds the RAB35 trucks, which keeps the antennas on the surface. But why is this specific assembly line running so incredibly hot in lupus patients in the first place? What triggers the factory to speed up?
Well, to understand the trigger, we have to look at the spark that lights the fire. TFA cells are unique among immune cells because they require persistent, ongoing stimulation through their T cell receptor, or TCR.
So the TCR is like the main ignition switch on the outside of the cell. When it detects an antigen like a target, it turns on.
Exactly. In a normal viral infection, the virus is eventually cleared, the target disappears, and the TCR ignition switch turns off. The cell stands down.
Right.
But in lupus, the antigen is you. It's your own DNA or your own cellular proteins. The target never disappears. So that TCR ignition switch is held down continuously, 247.
Oh, wow.
Yeah, and this sends a massive unrelenting signal deep into the nucleus of the cell to turn on specific transcription factors.
Okay, here's where it gets really interesting. Transcription factors are basically the genetic foreman of the factory. They read the DNA blueprints and scream at the cell to start manufacturing specific proteins.
Exactly. And the researchers identified specific foremen inside the nucleus driving this lupus process. The continuous alarm from the TCR activates formin that directly control our assembly line.
And we can actually use a little mnemonic here, right? Like C, SE.
Yeah, that works perfectly. So the S is SREBP2. That formin yells to produce more HMGCR, kicking off the whole MVA pathway.
Okay, so that's the start of the factory.
Right. Then the first E is at EGR1. That formin demands more GGPS1 to manufacture more of those GGPP screws.
No more screws.
And the final E is E2F1. That formin yells to produce more RE35 recycling trucks.
So the chronic stimulation from the autoimmune environment is literally demanding a massive upregulation of every single component of this recycling loop.
It is a totally vicious cycle. The hyperactive TCR signaling demands more GGTP and more recycling trucks, which keeps more and more CXCR5 antennas plastered on the surface, making the cell even more hyperactive and sensitive.
And they didn't just show this in a petri dish with isolated cells, right?
Right.
They looked at circulating CD4 plus T cells from human patients currently battling active systemic lupus erythematosus.
Yes, and the clinical data from the human cohorts is striking. In the blood of these lupus patients, they found significantly elevated levels of these specific factory proteins compared to healthy controls.
Wow.
And the expression of these assembly line genes positively correlated with the SLE disease activity index.
So the higher the patient's score, meaning the sicker they were and the more their organs were being attacked, the hotter the specific cellular factory was running.
This proves we aren't just looking at a quirk of mouse biology or an artifact of a lab experiment. This hyperactive MVA to RAB35 pathway is actively driving human disease severity. The formin are screaming to build more trucks, faster and faster, keeping the rogue commanding officers fully operational.
Which brings us to the ultimate question. If you have a biological factory where the alarm is stuck on and the formin are stuck in overdrive building these recycling trucks, how do you stop it?
You cut the power.
You cut the power. And the way to cut the power might already be sitting in millions of medicine cabinets around the world.
Yeah, the pharmacological fix theoretically exists in the form of statins. Statins are designed to block HMGCR, the enzyme, at the very beginning of the MVA pathway.
Right.
By blocking the start of the pathway, you inherently starve the cell of the downstream byproduct, the GGPP screws.
And the researchers tested 7 different commercially available statins, and here's a massive finding from the paper. Not all statins are equal when it comes to this specific immune-suppressing effect.
This is a vital detail for anyone interested in pharmacology or drug repurposing. The researchers evaluated the drugs using 2 key metrics, the IC50 and the Cmax.
Okay, let's break those down.
Sure. The IC50 is the half-maximal inhibitory concentration. Basically, it's the exact dose required in a controlled lab setting to shut down 50% of the rogue TFH cells. A lower IC50 means the drug is highly potent. You only need a tiny amount to break the factory.
But a drug that works in a petri dish isn't necessarily useful to a human being. That's where Cmax comes in, right?
Exactly.
Cmax is the maximum concentration that the drug actually reaches in a human patient's bloodstream when they take a standard, clinically approved, safe dose.
Right, and the math here dictates reality. For a drug to be useful in the real world, for shutting down these rogue immune cells, its Cmax, what's actually circulating in your blood safely, has to be higher than its IC50, the dose required to stall the recycling trucks.
Makes sense.
Out of the 7 statins tested, including incredibly famous ones like atorvastatin and rosuvastatin, only 1 made the cut.
Only pitavastatin. It had an IC50 low enough to work at standard human doses. Its peak serum concentration safely exceeds the threshold needed to suppress these cells.
Yeah, the other statins might work in a lab, but you'd have to give a patient toxic, dangerously high doses to reach the required concentration in their actual lymph nodes.
Right, which you obviously can't do.
Exactly. Pitavastatin's unique chemical structure allows it to penetrate these specific immune cells and inhibit the pathway at incredibly low concentrations.
And they proved this efficacy in vivo using 2 different animal models, right?
Yes. They used a lupus-like mouse model called BM12, which mimics chronic graft versus host disease. And they also used a spontaneous lupus model called MRLLPR, which naturally develops severe, fatal autoimmunity over time.
And when they treated these sick mice with pitavastatin, it didn't just tweak a few lab numbers. The results were dramatic. It significantly reduced the dangerous autoantibodies, specifically anti-dsDNA and ANA circulating in their blood.
Which is huge.
Yeah. It reduced inflammation in the kidneys, which is one of the primary causes of mortality in human lupus, and it actively cleared up their skin pathology.
Because the pitavastatin successfully starved the cells of GGPP, which stall the RAB35 trucks. This effectively stripped the CXCR5 GPS antennas off the surface of the rogue TFA cells.
And importantly, it wasn't just CXCR5, right?
No, it stripped other critical co-stimulatory surface receptors too, like PD-1 and ICS. It completely crippled the rogue officer's ability to communicate, navigate, and coordinate the autoimmune attack.
So what does this all mean? It sounds like we found a miracle cure hidden in a cholesterol drug. But there is a dietary plot twist in this paper that is absolutely wild.
Yeah, science rarely gives us straightforward miracles without a catch.
Right.
In the lab, the researchers performed a classic rescue experiment. They treated the hyperactive TFA cells with the pitavastatin, which successfully blocked the factory and stalled the recycling trucks. The cells went blind, just as expected.
Okay.
But then they manually added a precursor to GGPP, a molecule called GGOH, directly into the environment.
And instantly, the drug's effect was totally reversed.
Completely.
The cellular factory grabbed that precursor, the mechanics started turning out screws again, the recycling truck started up, the antennas went back to the surface, and the rogue cells came back online. The statin was rendered entirely useless.
It bypassed the blockade entirely, because the statin blocks the cell's internal production pathway. But the cell has a back door. If the precursor molecule is floating around outside the cell, it can just absorb it and keep the factory running.
And this is the kicker, because the researchers point out that GGPP and its precursors aren't just synthesized inside your body. You consume them in your diet.
You do.
They are highly concentrated in certain foods. Beef and soybeans are absolutely packed with these molecules. If you eat a leafy salad, you're getting 0 GGPP. But a steak or a soy burger?
The implication for patient care is staggering. Eating a diet very high in beef or soy could literally introduce enough exogenous GGPP directly into your bloodstream to completely counteract the autoimmune benefits of the statin.
That is insane.
The drug might be doing its job perfectly, blocking the internal factory, but your diet is actively smuggling the microscopic screws in through the back door.
It makes you look at autoimmune therapy completely differently. A patient could be taking the right drug at the right dose, but if their diet is flooding their system with the exact molecule the drug is trying to eliminate, they won't get better.
Exactly. It fundamentally changes how we might have to combine dietary restrictions with pharmacology. It emphasizes that we can no longer view medication and diet in silos. The biochemical environment of the gut and the blood directly dictates whether these rogue immune cells have the building blocks they need to sustain their civil war.
Okay, so if we tie all these threads together into a quick summary for you listening, the rogue TFF cells driving lupus rely on a constant supply of GGPP to keep their recycling trucks running and their antennas on the surface. The chronic autoimmune environment keeps this factory running hot. Pitavastatin can safely cut off the internal supply line, blinding the rogue cells and stopping the disease, unless your diet undoes the drug's hard work by providing a separate supply line.
That's the perfect summary.
It fundamentally shifts how we view standard medications. A cholesterol drug isn't just acting as plumbing for your heart, clearing out plaque. It is actively altering the microscopic recycling trucks inside your immune cells.
It is a massive paradigm shift for drug repurposing. But, looking at the entirety of this mechanism, it raises an important question.
Oh.
If statins successfully suppress TF cells by starving them of these screws, what happens when we actually need those TF cells to do their jobs?
Oh, right.
Remember, we need them to act as drill sergeants for the B cells to respond to vaccines. The researchers noted in their discussion that widespread statin use in the general population is actually associated with lower efficacy for the flu vaccine in older adults.
Whoa. Wait, really? So if you are taking a statin purely for your cardiovascular health, are you accidentally blinding the very immune cells you need to build immunity to the flu?
Based on this mechanism, it is entirely possible. The statin is starving the healthy TFA cells of GGPP, stalling their recycling trucks, and preventing them from navigating to the B cells to help make flu antibodies.
That is wild.
And if that's true, it leads to a rather provocative thought experiment.
Okay, lay it on me.
If dietary GGPP from beef or soy can override the statin and turn the cells back on, could eating a pre-vaccine hamburger rich in dietary GGPP actually help your immune system temporarily overcome your heart medication and make your flu vaccine work better?
Okay, my mind is officially blown. A daily cholesterol drug subtly disabling your immune troops, And a fast food burger potentially turning them back on just in time for flu season.
All connected.
We started this deep dive talking about the immune system as a highly trained military. It turns out, whether those troops are fighting a foreign enemy or fighting you, they truly march on their stomachs, or at least on their dietary supply of microscopic screws. I highly encourage you to ponder this incredible intersection of diet, cellular recycling, and your own immune system. Thank you for joining us on this deep dive.